An important virus-based vector is phage lambda vectors. Fred Blattner and his colleagues constructed the first phage vectors by modifying the well-known lambda phage. They took out the region in the middle of the phage DNA but retained the genes needed for phage replication. The missing phage genes could then be replaced with foreign DNA. Blattner named these vectors as Charon phages, after Charon, the boatman on the river Styx in classical mythology. In general, two types of lambda phage vectors have been developed, and these are lambda insertion vectors and lambda replacement vectors. One clear advantage of the lambda phages over plasmid vectors is that they can accommodate much more foreign DNA. For example, Charon 4 can accept up to about 20 kb of foreign DNA. Lambda insertion vectors accept less DNA than the replacement vectors, since the foreign DNA is simply inserted into a region of the phage genome with appropriate restriction sites. Common examples are lambda GT10 and lambda Charon 16A. In a replacement vector, a central region of DNA not essential for lytic growth is removed, which is called a stuffer fragment, by a double digestion with, for example, EcoR1 and BAM-H1. तो पूरे DNA में से सेंट्रल रीजन जो है वो रिमूव कर दिया गया, जिसे स्टफर फ्रैगमेंट कहते हैं. This leaves two DNA fragments called right and left arm. The central stuffer fragment is replaced by inserting foreign DNA between the arms to form a functional recombinant lambda phage. ये पूरा जो है वो intact DNA बन जाएगा. The most notable example of lambda replacement vectors are lambda EMBL and lambda ZAP. Lambda ZAP is a commercially produced cloning vector which includes unique cloning sites clustered into a multiple cloning site, which is MCS, as you saw in case of PBR, PUC, there are multiple cloning sites. So here in lambda ZAP is also a multiple cloning site present. Furthermore, the MCS is located within a lac Z region, providing a blue-white screening system based on insertional inactivation. तो जैसे आपने पहले देखा, लैक Z जो है वो MCS का पार्ट है, और इस लैक Z की प्रेजेंस की वजह से बीटा ग्लैक्टोसिडेज प्रोड्यूस होता है, जो कि एक्स घोल को एक इनएक्टिव ब्लू कलर में कन्वर्ट करता है. तो इस तरह से आप ब्लू वाइट सिलेक्शन की बेसिस पे जो है वो रिकम्बिनेंट कॉलोनीज़ को सिलेक्ट कर सकते हैं. सेम इस लैम्डा जैप में भी आप ब्लू वाइट सिलेक्शन कर सकते हैं. It is also possible to express foreign cloned DNA from this vector. This is a very useful feature of some lambda vectors, since it is then possible to screen for protein product rather than the DNA inserted into the vector. This screening is undertaken with antibody probes directed against the protein of interest. Another feature that makes this a useful cloning vector is the ability to produce RNA transcripts, which are termed as cRNA or riboprobes. This is possible because two promoters for RNA polymerase enzymes exist in the vector. One is a T7 and the other one is a T3 promoter, which flank the MCS site. One of the most useful features of lambda ZAP is that it has been designed to allow automatic in vivo excision of a small 2.9 kb colony producing vector, the phagemid P blue script SK. This technique is sometimes called single-stranded DNA rescue and occurs as a result of a superinfection process where helper phages are added to the cells, which are grown to an additional period of approximately 4 hours. A common use of lambda replacement vectors is in constructing genomic libraries. Suppose we wanted to clone the entire human genome. This would obviously require a great many clones. So the larger the insert in each clone, the fewer the total clones would be needed. क्योंकि एक क्लोन में अगर आप बड़ा डीएनए इंसर्ट कर सकते हैं तो टोटल क्लोन्स आपको जितने चाहिए होंगे जाहिर है वो कम होंगे, और अगर आप जो है वो छोटा इंसर्ट जो है, छोटे साइज़ का इंसर्ट जो है, फ्रेगमेंट जो है वो इंसर्ट कर रहे हैं तो आपको ज़्यादा क्लोन चाहिए होंगे. तो इस तरह से बड़े बड़े जीनोम्स को इन वेक्टर्स की मदद से क्लोन मैप किया जा सकता है. In fact, such genomic libraries have been constructed for the human genome and for genomes of a variety of other organisms.